Today we will be starting a new chapter known as Animal Kingdom. It is the fourth chapter in class 11th NCRT, and today we will be talking about the basics of Animal Kingdom, and in the next video we will be talking about the first three phyla, which is Porifera, Cnidaria and Ctenophora. So let's start with the basics. We have OSCSDCR. This is very important. OSCSDCR. Alright, say it with me. OSCSDCR. OSCSDCR. This is very important. So you have to memorize this. So O stands for organization. We have 4 types of organization. So all of these are used to, these are criterion. We can call them as criterion on or the basis on how we classify these organisms. Now we are talking about animals. So these are the basis on how we classify these organisms or these animals. So the first is organization. We have cell level, cellular level, which is found in sponges, also known as Porifera. We have tissue level, which is only found in Cnidaria and Ctenophora. Cnidaria is also known as Coelenterata. The third one is organ system, which is found in Platyhelminths. So cell system, the cellular organization, means that the cells, there are only one type of cells and there are a cluster of cells. So there is a cell aggregate. So it is also known as a cell aggregate plan. Next up is tissue level, where the animals have different kinds of cells. Then we have organ level, in which there are different new organs, and then we have organ system level, where there are like fully functional organ systems. So organ system, organ level of organization is found in platyhelminths, also known as flatworms. We will be talking about that in the third video after this. So then we have the organ system level of organization, which is first found in Aschelmanthus, and it goes all the way to chordates, where we are humans, we are chordates. So next up we have symmetry, a really easy concept. So in symmetry there are 3 types. There is asymmetrical, there is radial symmetry, then we have the third one, which is bilateral symmetry. So bilateral symmetry is found in us. Alright, radial symmetry is only found in Echinodermata, Ctenophora and Cnidaria. So what is radial symmetry, asymmetrical and bilateral symmetry? So first up, asymmetrical states that any organism such as Porifera, which is shaped like this is a rough representation of Porifera. So it cannot be cut into 2 equal halves no matter which plane you cut it in. Alright. But radial symmetry states that, for example, in jellyfish, if we take the top half, which is like this, it looks like this. Alright, so the top half of the top part of the jellyfish, if we look at it from above, we can see that any plane can cut it into 2 equal parts, like a pizza. And then there is bilateral symmetry. For example, us humans, and alright, let's take an example of us. Alright, so this is a guy, his name is John. Alright, so if we cut him from here, he is perfectly divided into left and right. Left and right. Oh, why is it bugging like that? Alright. Okay, so we can only cut him from left to right. Left and right. Alright, but we cannot cut him in any other plane which gives us that. So there is only one plane which can divide it into two equal parts, which is left and right. So that is bilateral symmetry. And the only asymmetrical is porifera. Then we go on to coelom. So this concept is a little bit tricky. Let me decrease the size of this. Yeah, that's good. Alright. So, coelom. There are two main types. There is diploblastic and triploblastic. So diploblastic states that there are only two germinal layers. So if you look at your textbook on page 39, 38 and 39, you can see that in figure 4.2a, the middle layer, the red layer, does not have any cells. So it is an undifferentiated layer, which is, so the middle layer is undifferentiated. And is also known as mesoglea. But in triploblastic, it has three germinal layers. So these layers are known as germ layers. The first one is the ectoderm. The second one is endoderm. The innermost one, and the middle one is known as the mesoderm, from which the notochord is derived. We will talk about this later. So triploblastic have three germ layers, which is the endoderm, ectoderm, and the mesoderm, whereas diploblastic only has the endoderm and ectoderm and has a mesoglea. So in triploblastic, there are three more types. Alright, so in triploblastic, there are three types. Again, there is the first one, which is acoelomate. Then there is pseudocoelomate. Then there is true coelomate. Alright, so acoelomate, as you can see in figure 4.3c, it does not have any cavity. So the coelom, the body cavity is lined by this mesoderm in our body. So this is known as coelom, the body cavity. But over here we can see that it does not have any body cavity. Everything is red, and there is a red circle in the middle layer. Whereas in figure B and figure A, we have some body cavity present. So these are known as acoelomates. The first one, which is an acoelomate, which is triploblastic, is platyhelminths. The first pseudocoelomate is Aschelminths. And the first true coelomate is Annelida. Alright, so pseudocoelomates the body cavity is not lined by the mesoderm. As you can see in figure B, it isn't lined by the mesoderm. It is just present in pouches, round pouches like this. But the body cavity isn't lined by it. Whereas in figure A, we can see the body cavity is lined like this by the mesoderm. And we can see that there is a line that comes in the middle over here. We can see a line over here. So that states that they are true coelomates because their body cavity is lined by the mesoderm. So they are true coelomates. So then we have segmentation. In segmentation, we have metamerism. So segmentation is where an animal is divided externally and or internally into segments. So they have segments and they have repetition of these. For example, we have earthworms. So if we take an earthworm, then we can see it has these round things like this. So this is known as metameres. And the phenomenon of these metameres is known as metamerism. So it is when the body is externally and internally divided into segments and has repetition of some organs. For example, we have earthworm, also known as Pheretima. Segmentation is pretty easy. Now we have the digestive system. So in digestive system, let's take a fresh new page. We have two main types. We have the we have the incomplete and we have the complete. So in the incomplete, we have only one hole, we have only one opening. For example, in Hydra, let's say this is the figure of the Hydra and these are the tentacles of the Hydra. Then we can see that it has only one opening, which serves as both mouth and anus. Alright, so that means the food goes in, it gets digested, and the waste goes out, the undigested food. Then we have complete digestive system. Let's again take the example of earthworm. So an earthworm, if we look at it, if we dissect it, it has sort of this kind of structure. So it has a mouth and a separate anus, which is separate. So this states that it has a complete digestive system. Alright, so this is Hydra. Alright, so then we go on to circulatory system. So there are two types again in circulatory system. So there is open type and there is closed type. So there is open and closed. So in open type, the blood, there are no tissues, there are no vessels which is present. So that means the blood is directly pumped into the body. So the tissues and all the cells are directly bathed in the blood. So the cells and tissues are directly bathed in the blood. For example, let's take a housefly. I know this does not look like a housefly. We have its wings over here and its little eyes. Alright, so inside the body, it has a heart present over here and like this. And so the deoxygenated blood goes from here and it goes through the heart where it gets oxygenated and then immediately gets bathed into the whole body over here. Then it goes again over here in order to get oxygenated and exits over here. So that is the fly. So that is an open digestive system. Then we have the closed digestive system. Let's take us as an example. We have a closed digestive system because we have vessels. We have blood vessels. Right, we have blood vessels, arteries, veins and capillaries. There are three main, alright? Three main vessels. Very important. So there are arteries, veins and capillaries. Alright, so we have arteries, veins and capillaries. So then we go on to respiratory system. So there isn't much in respiratory system. So if we go on to page, I think 47, 44, we can see that there are different types of respiratory system. So we have book gills, we have book lungs, gills. Oh my god. Alright, so we have the gills and we have the tracheal system. So these are the four types. And now we move on. Oh no, this is the end of this video. In the next video we'll be talking about Porifera, Cnidaria and Ctenophora. So this is the main basics. So let's just go over what we learned today. So let's use this. Okay, alright, this is pretty cool. I'll use this blue color. Alright, so we have the animal kingdom, in which we have OSCSDCR. Very important. Alright, OSCSDCR. OSCSDCR. This is very, very important. I'll give this 5 stars. 6 stars out of 5 stars. It is very important. Alright, so O stands for organization, S stands for symmetry, then we have coelom, segmentation, digestive, circulatory and respiratory. So these are the criterion that we use for classifying organisms. Then in organization we have cellular level, tissue level, organ level and organ system level of organization. The first one to have organ system is Askelman's and V chordates all the way to chordates. Alright, the first one to have organ system and the only one is Platyhelminths. Then we have Cnidaria and Ctenophora. These are also known as brothers because they are nearly identical. They are very, very similar. So they are known as brothers. Then we have cell level where only sponges are present in the cellular level of organization. Then we have symmetry where we have asymmetrical, radial and bilateral. So in asymmetrical, no plane can dissect. In radial symmetry, there are multiple planes which can dissect an organism. In bilateral, there is only one plane that can dissect in left and right. Alright. Then we have the coelom. We have diploblastic and triploblastic. diploplastic means that they have only two germ layers, which is the endoderm and ectoderm, and they have the mesoglea, which is the undifferentiated layer. Then in triploplastic, we have three germinal layers, which is the endoderm, ectoderm, and mesoderm. So we have the acoelomates, the pseudocoelomates, and the true coelomates. In acoelomates, there isn't any body cavity present. In pseudocoelomates, there is a body cavity present, but the body cavity isn't lined by the mesoderm. The mesoderm is present in scattered pouches. Then in true coelomate, we have in true coelomates, the body cavity is lined by mesoderm. Then we have segmentation, where it's just the repetition of organs. So it is the externally and internal division and repetition of organs. So for example, in earthworm, also known as Pheretima, we have segmentation. Then we have the digestive system. We have an incomplete and complete digestive system. Incomplete states that there is only one hole, alright, which serves as both mouth and anus. Then we have the complete digestive system, which states that there are two holes, the mouth and the anus. So this is a complete digestive system. So then we have the circulatory system. We have an open and closed circulatory system. Open means that there are no blood vessels. The tissues and cells are directly bathed in the blood. Then we have the closed digestive system, where we have blood vessels. Alright, we have the three main vessels: arteries, veins, and capillaries. Alright, so that is the end of today's video. Thank you so much for watching, and be sure to check out the next video, in which we will be talking about the three phyla, which is Porifera, Cnidaria, and Coelenterata. Porifera, Cnidaria, and Ctenophora. And then in the next video after that, we will be talking about Platyhelminths, Aschelminths, and Annelida. Thank you.